Hey, good morning, everybody. We are getting ready to resume our morning session for the Giovanni workshop. Our second speaker in this morning's session is Dr. Adnan Al-Azri, who is an associate professor at Sultan Qaboos University in Oman. Adnan?
Thank you, Jennifer. Let me start by really thanking NASA and Giovanni team for really giving me the opportunity to speak to today. Before I do that, and I would like really to say hello to everyone. Good morning or good evening, depends where you are. Today I'm going to share with you some of the aspects of the content in our paper that we published this year, the mesoscale and nutrient condition associated with the massive Cochlodinium polykrikoides in the Sea of Oman and Arabian Gulf. Um, uh, in order really to give you a full picture, I would like to start by giving you the background information for people probably who have no idea about the area. How I'm going to do that is by um, looking in aspects or forces behind um, this productivity. I'll be looking at the varying physical and chemical forces and looking at the challenges that we have in Omani water, and also looking at the biological response in the Sea of Oman and Arabian Sea, and link these um, two basins and see how they differ and what really are the forces which ended up causing the problem that as I'm going to focus more to the end of my presentation, which is the outbreak of Cochlodinium in 2008-2009, and ended up with wrapping up the summary of my talk. So for people who have no idea where we are, Oman is here, and it has an extensive coastline of 3,165 from north all the way to south. The biological characteristic of the ecosystem of the Sea of Oman are influenced by hydrodynamics of the area, driven mostly by monsoonal winds. I'm going to really to spend more time on this, explaining what are and when they are happening, and the outflow of water from the Arabian Gulf and the inflow from the Sea of Oman to Arabian Gulf. Just to give you an idea, during a typical year, you find that the southwest monsoon occurs from June to September and creates coastal upwelling um, in the south here. And during the northeast monsoon, eddies, as we can see later, the formation of eddies in the Sea of Oman and the influence of the low oxygen water in the Sea of Oman, and also the outflow of warm water from the Arabian Gulf. So, speaking of um, the monsoonal winds, what they affect, as I said, during the southwest monsoon, um, the nutrients which are coming from the result of the upwelling in the south of the country, it does inject some nutrients which cause productivity offshore the Arabian Sea and cause extreme phytoplankton biomass. And we've been experiencing or observing this high biomass. And over decades, this biomass showed a shift in species composition from diatom to dino, basically to Noctiluca. And Noctiluca has been one of the species that has been really occurring in Arabian Sea and Sea of Oman for more than a decade now. But in 2008, 2009, this was um, dominated by Cochlodinium polykrikoides. So that's why we wanted to understand why this event happened, the shift from Leptoukh Giovanni to Cochlodinium polykrikoides. So in order to have a clear pictures of the reversal of the monsoon winds, I will go now to a simulation, which probably will make a picture even more clear using the NRL model. So as you can see here, the upwelling I was talking about here, it happens during summer, and let me just get the so you can see the south of the country. During the summer month, starting from June, there is an upwelling taking place, injecting water offshore the Arabian Sea, and that's where the blooms usually start really forming and moving north to the Sea of Oman. And during the northeast monsoon, you start getting the propagation of this area in the Sea of Oman, and you starting getting high productivity, and these blooms move inshore and starting causing all kind of problem. So as I'm progressing, I will be explaining exactly in more details what are the mechanisms, what the impact to these blooms due to the marine resources in the coastal water of Oman. So these are the eddies I was talking about during southwest monsoon and northeast monsoon. As you can see, we have two types, the anticyclonic eddies and the cyclonic eddies, which are more productive in comparison to anticyclonic. This eddy really do play a big role in distributing the nutrients and even distributing the bloom themselves, and depending on how the time they spend in the Sea of Oman, the impact of this bloom in terms of fish kill or causing other problems, it all depends on how long and which direction this eddy are propagating in the Sea of Oman or in Arabian Sea. So with this oceanographic setting in the Sea of Oman in Arabian Sea, we do have a lot of challenges. Dr. Pavel mentioned about the dust. This is one of the issues also, that we have no clue how much does it influence the productivity, for instance, in Arabian Sea. So just to name a few, as you well aware, this is a region where there is high oil production, and as a result, is the world #1 region in terms of oil tanker traffic. So we are vulnerable to all kind of problem in terms of either oil pollution itself or the ballast water in terms of invasive species. And if I can also mention the lower image here, showing the desalination plant in the Arabian Gulf, and the same story happens in Oman. We are in arid or semi-arid region where we don't have a good source of fresh water, so we rely on desalination plant. So with the expansion in population, we require more and more of this desalination plant along the coast. And since 2007, we've been hit by a massive cyclones in 2007 and did a lot of damage in the marine environment, and the extent of this damage still not clear for us. So there is a lot of challenges that we do have, but this is not my topic today. It's just to give you an idea what are the challenges we have. Although that my focus will be on two species, Cochlodinium polykrikoides and Leptoukh Giovanni, we do have a long list of other species, dinos and diatoms, which are potential to cause problem in terms of fish kill, even to human health, which we're still investigating. So as I said, my focus is going to be on these two species, and these are not just limited to Oman. They are well known that they are limited. Um, um, it's a worldwide group that found in many places in the world, and the species that we have in Oman is closely to what is found, the species is found in the States, in Malaysia, and Puerto Rico. And when we talk about Noctiluca, we have two types, the red Noctiluca and the green Noctiluca. And as you can see, the red Noctiluca, it's more geographic distributed more than the green Noctiluca. And the green Noctiluca, you can see, is more or less in this region. Although that there have been reports that it also occurs, for instance, in eastern Arabian Sea, and even in Oman, there's some reports, in mid 90s or late 90s saying that they were red Noctiluca. This raises a number of questions when we start talking about, for instance, the red Noctiluca and the green Noctiluca. In a sense that the image that you see, the left upper hand side image, this picture was taken in Arabian Sea, in the middle of Arabian Sea. So this is a good sign. As I mentioned earlier, during the southwest monsoon, nutrients are injected offshore the Arabian Sea, and that's where this bloom manifests, and they are carried by the mesoscale eddies into Sea of Oman. So what I want to mention here is the issue of red and the green Noctiluca. If you see in the upper left image, you could see that it's green, and if you look under the microscope, it's still low green, and the reason behind it is the symbiont species. This species is why is this successful? If you look very quickly in its physiological or ecological characteristic, the Noctiluca it's known as the to exhibit a phagotrophic, meaning they can rely on a symbiont, or they can graze on diatoms, copepods, eggs, and even some small larvae. So and it has the capability of migrating up and down in a water column. When it comes to its color, when it's approaching the shore, either you find it in a mix of brownish or orange, and this still posing many questions. What exactly what is red and what is green? Because when we look under the microscope, when the cell is in the later stage, when it's dying, it's changing its color from green to brownish, and when it releases the Pedidomonas noctiluca, the symbiont which lives within the cell itself. And what we have in the right up images, which is pure orange, it happened the beginning of this year, where we found that rather than having only the symbiont species, we found that it was full of diatoms and dinos, which probably reflected the orange color. And if we look at the Cochlodinium, it has also some of characteristic which allow to survive and to dominate in other group of phytoplankton. Just to mention a few, there are a number of research that really reported that this species it's eurythermal, euryhaline, meaning that it can live in a wide range of temperature changes and salinity. And it has the it can form a cyst which can really survive harsh condition and germinate when conditions are much better. So just to give you just an idea, so when moving now focus on this species, the first thing we noticed when we heard about this species started from the north of the country, and we thought that because people are saying that we have a brownish water, we thought that this is the normal Noctiluca, which we're having every winter, greenish or brownish color. So then we started having complaint that the massive foam along the beach. And then we starting hearing report that people were really annoyed with the strong odor which is coming. So we knew that this time we are dealing with a totally different species. And then when we took the sample under the light microscope, we start noticing that yes, this is totally a different species we're dealing with. But we thought it was Gymnodinium catenatum until we had the SEM microscopy to look very closely. And then it was identified as Cochlodinium polykrikoides. So when it comes to impact of this species, is very interesting in a sense that there's still a great debate now. What is the mechanism of this species in terms of fish kill? There are people suggesting that suffocations and because of the reactive oxygen, which caused osmoregulatory alteration. And people suggesting that this is basically it kills the cage fish in the cages, but this was not the case in our water. So it was to extend that, as you can see, there was a massive fish kill, as reported by the Minister of Agriculture and Fisheries, about 200 tons, and this was distributed into in the fish in cages and wild fish and even shellfish. And its impact, it went beyond a fish kill. It affected the salination plants because they were clogged with the massive blooms. And refinery, they couldn't get the proper water because of the presence of this species. And not to talk about the coral reef, which really damaged. And some schools complain about strong odor and were worried about the kids to be really affected. And according to one of the papers, they reported that some of the schools were closed during that period. So this is just a background, to project the image. So if we focus about the paper that we publish, what exactly was the motive? Because we're dealing with the different issues, and that's where really we were very thankful with the NASA and Giovanni team. There is another tool that's helped us a lot, especially during this event, really to understand what was happening in a broader scale. Because our time series, you're going to see, is very coastal, and we didn't have a proper research vessel to go far offshore to these areas and really to do the sampling. So the motive or the objective was to determine the physiochemical mechanism leading to the unprecedented outbreak of these blooms. And once we understand, then because during the bloom itself, we were really bombarded with a lot of questions in terms of what it is, what we can do, and how we can get rid of it. And people thought it's a very simple thing that we can just dump something or do something and it's done in 1 week. That's why we look at it very, very closely in order to come up with a proper idea of this outbreak in order to have decision maker to come up with a strategy for mitigating this species and also other group in the future. So our data sampling, so we had in situ measurement where we had a salinity temperatures of oxygen, phytoplankton taxonomy, and chlorophyll nutrients. Some of the parameters, we use the CTD, and we use also analysis in lab, for instance, for chlorophyll and nutrients. And for satellite measurements, we use the topics, Poseidon JSON, altimetry, sea surface temperature, chlorophyll, time series analysis, visualization of satellite data, was undertaken with NASA Interactive Online Satellite Data Analysis to Giovanni. So to give you an idea where our station. So once we realized that this was almost a regional issue, it was very interesting at the beginning. Instead of coming together and understanding what was going on, I think we started pinpointing to each other. I think this is coming from the Sea of Oman, this is coming from the coast of Iran, and this is coming from the Arabian Gulf. But sooner or later, we realized that since we didn't know exactly what was happening, we decided to have a small workshop that we discussed and really try to understand what was the problem and what was each side of these three basins were experiencing. So this is our stations here, and then what you see here is from Iran side, that where the scientists following closely, and the station in United Arab Emirates. The first thing we noticed when we're looking at the data, it was a drop of the temperature. I hope you can see the cursor because I'm trying to drag it, but I cannot see it myself. I don't know what's the problem. But I hope that as you can see from July on the lower figure, there was a drop of almost -3 degrees unusual temperature during summer. And this is most of the time it's the result of the southwest monsoon upwelling water which are injected in the Sea of Oman. And the next thing that we realize is the sea surface height anomalies, where as you see, as again, I'm not sure if you see my cursor because I cannot see it. If you look at the trend, you see that towards the end of the graph that during this period we have the highest anomaly and the lower negative, indicating the persistence of the northeast monsoon. So what does it mean? This means that having the strong upwelling in the south and having as we progress from July to all the way towards November, you could see that bloom starting developing almost in November. As reported by the Iranian side, there was a strong upwelling which kind of just supports this bloom even to move south, and the outflow from the Arabian Gulf towards the Sea of Oman. So as we progress, these blooms move southward. That's where we're starting our sampling and understanding exactly and taking different measurements in order to understand the progression. So if before you want to go any further for the progression, if we start looking at what was happening in terms of nutrients, it was very interesting to see the, for instance, ammonia. What you see in the upper graph, that's level of ammonia. We have a station which we've been monitoring for the last 8 years, or time series. It hardly getting 1 or 2 micromole as the highest. But as you can see, during the bloom period, ammonia reached almost 12 or 15 micromole in some station. And the same applies to urea. And although that's nitrates, yes, we used to have a nitrate, but as you can see again, it was exceptional, and phosphate as well. And if I can zoom the area where during the bloom itself, it was very interesting. There was a sequence of having high ammonia at the beginning where the bloom was around, and starting really disappearing in urea, and then we're switching to the increase in terms of nitrates. And when we look at the red feed ratios, again, looking at the inorganic, it was very interesting to see that it was mostly below the red feed ratio of 16. And then while the organic showed very high, so as an indication that there must have been a high phosphate as well. And this is not new. That's all the places that reported the outbreak of this species. It kind of showing high phosphate as well, but we have no idea in this water where exactly this phosphate came from. And again, showing that the high ratio for organic nitrogen and phosphorus, that the organic nutrient did contribute. And this is true in a sense that when we were observing this bloom, we could see that their persistence for a few days and disappearing, and people were happy that it's over, but we knew that it's going to come up again. And then a few days later, it starts coming up again. So we kind of just comparing our ratio with other places where they had problem with this species, for instance, Chesapeake Bay, and Narragansett Bay, and Gulf of California. So we found that almost they're following the same pattern except the one point that we have here. So we knew that the behavior of this species, what has changed in terms of chemical component of the water. So we're right, because the reason I'm saying this is that when we started getting the first data, we're really frustrated because we're not used, for instance, to the chlorophyll of more than, let's say, 30 microgram. But during this bloom, it was happening. The high high in ammonia, high urea. So when we realized that this was happening in other places as well. So to give you an idea, the influence of the nutrients and the species itself, if you look at the temperature changes in abundance of Cochlodinium and other dinoflagellate and diatoms, it followed nicely the when at the beginning when we started our sampling, it was very clear that Cochlodinium polykrikoides was totally dominating as a monospecific group and couldn't see anything under microscope except Cochlodinium. As we progress with our sampling, we started seeing other dinoflagellate, and then toward the end, diatom starting showing up. And this was the same, just to give you an idea on the depth of this station. For instance, the Azeba, which is the top graph, because as I said, my cursor, I don't see it. Anyway, this is almost 15 meter, and the Fahal, which is the middle one, is almost a 25 meter, and the Mask is almost 35 meter. One of the things that we observe, we read in literature, they said this species can only migrate up to 20 meter, but we clearly see that it can go beyond that. So this is one of the other characteristic of this species that we observe during these blooms. So again, back to the satellite altimetry data, we knew that because of the mesoscale pattern of these two basins, Arabian Sea and Sea of Oman, this group, once they already there is an outbreak of this bloom, we knew that they're going to go anti-clockwise from Iranian side, passing through the towards the north of the country and going down all the way down to the Arabian Sea. So this bloom persisted for almost 8 to 9 months until the kickoff of the southwest monsoon. Then it disappeared. But a good question is all the places they reported the outbreak of Cochlodinium, it persisted in this water up to this moment. But in Oman, we did see a few cells, but not to the level of outbreak. So we know that sooner or later this is going to happen again. So again, the one thing that we know that this species caused really damage in our water, and we still have a lot to learn about this species, and its, as I mentioned, mechanism of kill still unknown, and it's very clear in the type of fish or organism that it killed during the outbreak of this bloom. So to bring together the scenario of what happened during this bloom, beginning of July, there was unusually strong westerly winds, stronger than normal upwelling along the northern Omani coast and the Strait of Hormuz. And this did help somehow to inject the inorganic nutrient, especially phosphate. And as we progress in October, there is a discharge from the Arabian Gulf towards the coast of Oman, and there was initiation of this species along the coast of UAE. And as we progress, organic nutrient kind of contributed in terms of regeneration from dead fish. That's give it even more required nutrient even to persist longer in the water. And then more blooms starting really showing up in the Sea of Oman. And again, as I showed earlier, there was more nutrient to support this bloom even to persist long as well in the coast of Oman. And not to forget that the mesoscale eddies and cyclonic anticyclone, they do play a big role in distributing these blooms in Sea of Oman and Arabian Sea. So along with the mixotrophy of this species and its capability to survive high temperature and high saline water, so I think that's why it survived for longer. But the observation that I want to make here is that during these blooms, the Noctiluca scintillant, which is a common species that we've been seeing for almost a decade happening during winter, it can vanish in the water. We saw very few cells in Arabian Sea, but not in the Sea of Oman. That again is a big question mark. What exactly? Um, and we know that Noctiluca as well is a species that can very survive in a harsh condition. So to conclude, harmful alga blooms are serious, and I think we're noticing that it's a growing threat really in our water, not just for fishery, but with a long list of potential toxic species. And the country now is going towards diversifying their national economy, going towards aquaculture and tourism. So I think there are more challenges to keep the balance and keep a very close eye on these harmful alga blooms. And honestly, the satellite data was a very helpful tool really to understand the progression of the blooms, especially in the two basins. And again, we do believe that developments in human activity and the unique oceanographic feature of the Sea of Oman Arabian Sea are responsible for this hub's expansion. And a question that probably is our mind that this shift in especially the outbreak of Cochlodinium, is it the first sign showing that our ecosystem is becoming really fragile and starting really could be shifted very easily? And last but not least, we just want to say that in-situ data we use together with NASA satellite really product, a Giovanni, offer a powerful decision support tool for mitigating losses due to harmful alga blooms in the coast of Oman. And I want really to acknowledge NASA and Giovanni team for really making the powerful tool as we see it in order to understand these harmful alga blooms in our water. Thank you very much.
Okay, so thank you, Dr. Al-Azri. What we will do next is we will move to the question and if anyone has any questions for Adnan, please type those into the Q&A pod here on the right-hand side of your screen. Any questions? And also just a note, I see I need to upload your presentation file as well, Adnan, to the pod, but I do have the animation loaded below, and I will go ahead and upload that file as well. Anybody have any questions? I will do that during the break. There must be a question. Anybody? And great job. Okay, so there was a comment, Dr. Al-Azri. It says, thank you for that intuitive presentation on algal bloom and effects of nutrients in our waters. The first question is, are there any other areas where Cochlodinium blooms have been reported?
Um, if if the question is focused in the region, there were no reports of this species in the region.
Okay, thank you for the answer. The next question is, how can low oxygen trigger a bloom?
Um, uh, it's not as a single factor, it's a multi-factor triggering, but we're mentioning low oxygen in terms of survival, and there have been paper published even recently showing that especially Noctiluca scintillans um, dominate as a dominant species in these blooms because they can easily survive in a low oxygen in comparison to other groups.
Okay, thank you. The next question is, could you speculate why the strong westerly winds took place that year?
I think this is a good question. Honestly, I have no idea.
Did you hear the question?
Yes, I did. What I was saying, I think, is a very good, very good question. But I'm sorry that I have no idea why we had
Okay,
that
thank
year
you.
was really.
Okay, I see that Dr. Acker is providing some sort of an answer here. He's typing into the pod. Okay, let's see. Okay. So it's actually more of a question. Do low oxygen conditions occur in warmer weather?
Yes, as I mentioned, the two pattern that we have with the reverse monsoon. There was a study which was done in Oman that showing during the northeast monsoon, the layer seems to really to move very close to the surface. So in general, our water, the oxygen, the minimum oxygen layer, do very easily move close to the surface. Basically like 10 meter.
Okay. Thank you, Dr. Al-Azri. Are there any further questions? I'm scrolling here just to determine whether or not there are. Okay, let's see. Here is give me just a moment. Okay. It is a comment question. I live in a country where most of our people rely on marine resources, but with rapid growth in population and high demand of fish and sea resources, we have exploited much of our marine environment. Algal bloom, low oxygen content in our waters, especially in near-coast regions, have depleted fish catch. Perhaps fertilizers and insecticides have reached the sea via river systems and estuaries. So I don't know if you are seeking a comment from Dr. Al-Azri on that input. Is that, I don't see a question there necessarily, but more so of an observation and comment. Okay, so the next question is, regarding winds we could investigate with MERA data. It's actually more so of a comment by Dr. Acker.
Okay.
Okay, and the next
Yeah, I'll be
question
very.
is, go ahead, Jim. I'm sorry, go ahead. Okay, the next question is, can you speculate on the source of the nitrogen and phosphates?
I think I can speculate on a source of nitrogen, but not phosphate. Nitrogen, there are area that we suspect that is coming from the industrial area, although that in some places they claim that what has been dumped into the sea is be treated, but we do believe that a part of it is coming from land.
Okay, thank you, Adnan. Let's see if there are any further questions here. Okay. So this is a follow-up to the individual regarding the conditions in his geographic region, and Dr. Acker has noted that he will address how to access data that could be of assistance for that type of analysis using Giovanni. Is that correct, Jim? Yes. Okay, great. So let's see. Any further questions? We've got about 5 more minutes left to the question and answer period. Are there any further questions for our speaker? Okay, let's see. Give it just a moment or two more. And if there are no further questions, I will go ahead and upload Dr. Al-Azri's presentation, and then we will have a 15-minute break, at which point we will resume at 11 o'clock Eastern Standard Time with our final speaker for this morning's session. Okay, so it's a general question, Adnan. Are there any analysis capabilities in Giovanni that would augment this research?
I think honestly, in our team, there's one of my colleague who's really specialized in this area, and in my area is more or less what is happening in terms of institute information. So I'll be very happy to bring him on a picture and really to follow up this conversation, find how really we can get further help from Giovanni team.
Okay, let's see here. Seems like it would have been helpful for your study if Giovanni supported station or point data.
I agree, because when we dealt with these blooms, we try really to sample very inshore, and then I said again, it was more or less, we just suddenly, knew about these blooms. So our focus was, our time series. So we knew that's okay. This is gonna do the anti-clockwise movement or distribution along our coast from the north to south. So let's see what's in it exactly. But I agree, if we could just really access the data and get some help to have one station that really has been getting in a more type of analysis along with what we're seeing in inshore, I think it could be very, very helpful.
Okay, thank you. We've got 2 more minutes remaining during the question and answer period. Any additional questions? Okay, we've got about a minute left to the Q&A. Okay, what we will do is we will move to the break period at this time. Right now I will upload Dr. Azri's presentation, and then we will take a 15-minute break. You can see I'm uploading his presentation currently. When that has completed upload, in order to download the files, for those of you who have just joined, all you do is highlight the file and the option to download that file will appear. So it should be uploaded in just a moment here. And then we will move to our 15-minute break. Okay, well, thank you so much, Adnan. I don't know, did you want to participate as a participant for the remainder of this afternoon? It's getting later, I know. part of the world, so I don't know if
Yeah, I
you're
think it's
signing off.
getting late. Yeah.
Are you
I'll be
signing off?
signing off. I think it's getting really late. Yeah, I'll be signing off after this. Thank you very
Okay, so
much,
I will
Jennifer, for your
forward to
support.
you any additional questions, okay? So Adonis, I will forward
Sure.
your final question to Dr. Al-Azri, okay? Does that sound fine?